Anyway, we are almost there. We'll get through what we need to get through this morning. We'll get through what we need to get through in lab today, and then all that's left to do is study for your final exams. That's exciting. All right, so without, I guess, any more blabbing on my part, let's go ahead and jump in. So on Wednesday, we had gone over the male reproductive tract. We had talked about the process of spermatogenesis and how in males, at least, spermatogenesis is about as textbook of meiosis as you can get. It doesn't start until puberty, and then we have that spermatogonium that undergoes mitosis. One of those daughter cells is going to enter meiosis. The other one stays in the seminiferous tubule to continue to undergo mitosis. And we end up with 4 gametes as a result of that mitosis. So we've gone over all of that on Wednesday. Today, what I wanted to do is I wanted to spend a little bit of time introducing the female reproductive tract. Obviously, in lab, we'll have a little bit of time to go over the models and stuff like that. I'll go into a few more of the nuances about what the models show and stuff like that in lab. But in order to talk about what I want to talk about today, which is sexual development, I kind of have to introduce this first. So we'll go through this really briefly, enough to where we can understand sexual development, and then what we'll do is we will talk about some of the nuances of this in lab today. All right. I guess that's one of the downsides of having lecture before lab is things aren't always perfectly timed, and I guess it is what it is. Okay, so let's go ahead and dive in then. So the female reproductive tract, in many ways, is like the male reproductive tract. At the most basic level, we have a gonad that is responsible for making gametes. And then we have a duct system that gets those gametes out of the body, right? So we have this kind of basic similarity between the two. But that's actually where a lot of the similarities end. Beyond that, we have some pretty stark differences. For starters, the gonads, the ovaries, are found inside of the abdominal pelvic cavity, right? We saw with the testes that they're found outside of the body cavity, and we said that is really just a temperature issue for sperm production. Ova or eggs can be produced at body temperature with no problems. So there's no real reason to keep the ovaries outside of the body. So they are found firmly in the abdominal pelvic cavity. The other, I guess, big difference that we see almost right away is in the male reproductive tract, we see that the epididymis is connected directly to the testes, right? The seminiferous tubules are continuous with the epididymis, so there's this continuous tube leading out of the body. In the female reproductive tract, we see that that's not actually the case. In fact, the ovaries are not directly connected to anything at all, okay? The egg is obviously going to hopefully go into the fallopian or the uterine tube, which is the tube that we see right here. But there's no direct connection between the ovary and that fallopian tube, right? In other words, they're not directly connected to each other. So how do we get the egg from the ovary once it's ovulated into the fallopian tube? Well, the best we can do is hope, and that's exactly what we do. We increase our chances by really kind of flaring out the opening of the fallopian tube. You'll notice that the opening of the fallopian tube is called the infundibulum. So what it does is it really flares out, and it tries to surround the ovary as much as possible. There are these finger-like projections coming off of the end of the fallopian tube called fimbriae. So the end of the fallopian tube is kind of doing this, and it's trying to surround the ovary, which is right in here. So that when an egg is ovulated, what it does is it tries to act like a little funnel and catch that egg and bring it into the fallopian tube. The fimbriae, those finger-like projections, kind of beat and wave like this to try to sweep the egg into the fallopian tube. And there are some chemicals that are given off that sort of work to attract the egg as well into the fallopian tube. But there's no guarantee that the egg will actually go in, okay? It really is just a game of chance that's being played here. 99.9 times out of 100, it works, right? The egg goes into the fallopian tube. But there is a very small percentage of the time where the egg doesn't actually get into the fallopian tube. That egg can actually attach to other tissues in the abdominal pelvic cavity. And when that happens, we talk about it being ectopic. That can happen if the egg is unfertilized. It can actually also happen with fertilized eggs as well. And when that happens, it's called an ectopic pregnancy, and that can be life-threatening to mom. So we have to terminate that as quickly as we possibly can. It also wouldn't be viable anyway. But most of the time this works. But we definitely see that there's no direct connection between the fallopian tubes and the ovaries, which is very, very different than what we see in the male anatomy, where it's this continuous tube from the testes out of the body. So we have the ovaries, we have the fallopian or the uterine tubes right here, and the egg, of course, would travel through this, and it would travel to the large organ of the female reproductive tract, which is the uterus. The uterus, of course, is where implantation would occur if pregnancy were to happen. And it's as you can see here, it's a really thick muscular organ. It is comprised of 3 separate layers. We have that inner layer of epithelial cells that we call the endometrium. We have an inner layer of muscle called the myometrium. What kind of muscle is it? Smooth muscle, right? It is smooth muscle, right? Smooth muscle, right? Remember, we're in our smooth muscle era. And then we have an outer layer of connective tissue called the perineum. Okay, so this is a thick organ. We'll talk a little bit more about the uterus and specifically about the endometrium a little bit later on today. The uterus is separated from the vagina by something called the cervix. We can see that that's right here. Okay, this is the cervix that we see right here, and it's that separator between the uterus and the vagina. And then, of course, the vagina leads to the external environment. So this is essentially what we have in terms of the female reproductive anatomy. Because it is all internalized in the abdominal pelvic cavity, we have to work to suspend it. So we see a fairly significant network of ligaments that sort of suspends it within the abdominal pelvic cavity. You'll notice right here, there's a really big sheet-like ligament right here. This is called the broad ligament. It obviously would attach to the abdominal pelvic wall here to help to suspend the fallopian tubes, to help to suspend the ovary, as well as the uterus itself. The ovary is attached to the fallopian, or to the uterus, by something called the ovarian ligament that we see right here. And then there's also a round ligament that also works to suspend the uterus as well. And in lab today, we'll be able to see those on our models really pretty clearly. And then finally, the ovary is also suspended by a suspensory ligament that kind of hangs up in this direction right here, that kind of helps that ovary to sort of, well, for lack of a better term, hang within the abdominal pelvic cavity. So a significant amount of connective tissue kind of holding this structure in place inside of the female abdominal pelvic cavity. Right? Again, we'll kind of revisit this today in the lab just a little bit, but but this is essentially what we have. Does anyone have any questions so far? Okay. So, one of the things I guess I'm a little bit remiss on, and I usually rely, I guess, on models in the lab, which is why I don't have it. But I don't have a slide of the external anatomy. But in terms of external anatomy, we have a couple of primary structures. We have the labia majora and labia minora, and then we have the clitoris itself, right? So we'll be able to take a little bit of a closer look at those today in lab. But that is sort of the external anatomy. Okay. So what might be helpful for us to do is, well, let's do it in a minute. I was going to make sort of an inventory of male and female internal and external anatomy, but let's hold off on that for just a second. So I'll let you guys just finish up if you have any, are taking any notes on this slide. Again, we'll return to this at the beginning of lab today, but I want to take a look now at sexual development.
Question?
Yes.
You said the uterus is separated from the vagina by the cervix?
Yes. Okay. Scooch around in my things here. Okay. So let's talk a little bit about sex determination and sexual development then. So one of the things that really sort of boggles my mind, especially the more I think about it, is that sex determination really depends upon the presence of the Y chromosome. Okay, and you may think to yourself, well, yeah, everybody knows that. Why does that boggle your mind? Sometimes I think a little bit too hard about things, but let me explain why it boggles my mind. The Y chromosome is kind of an interesting structure. Where am I at? Let me make sure I get this right. Let me talk about the Y chromosome before I talk about this. Okay, so the Y chromosome is interesting. It consists of only 27 genes that are specific to the Y chromosome. So in terms of chromosome size, the Y chromosome is, well, quite frankly, kind of puny. By contrast, the X chromosome has about 1,500 X-specific genes on it. So we're looking at a huge difference. Only 27 genes are specific to the Y chromosome. In terms of all of the genes that we have, that's an incredibly small fraction of genes. Okay. There's one gene, though, in particular on the Y chromosome, that accounts for almost all of the differences that we see between males and females. And that gene is called the SRY gene. Right? SRY. Okay. S stands for sex. It actually stands for sex determining. Sex determining region of the Y chromosome. Okay, so SRY stands for sex determining region of the Y chromosome. This one gene accounts for almost every single difference that we see between males and females. And I don't know about you, but that's astonishing to me that one gene can have that much power in terms of the way that we look, in terms of the way that we function, in terms of the way that we act, right? It's really pretty remarkable that one gene has that much pull over so much of our biology. Okay. For what it's worth, there are a lot of Y-specific genes on the Y chromosome that have absolutely nothing to do with maleness. One of those genes happens to code for tooth enamel, and another for a ribosomal protein. So a lot of them are inconsequential to our sexual development. But that SRY gene is really, really important. Kind of showing a picture of the two chromosomes next to each other, or at least a diagram of the two chromosomes next to each other, we see that there is this massive size difference between the X and the Y chromosomes. The X-specific region, as I mentioned, has a little bit over 1,400 genes on it, whereas the Y-specific region only has 27 genes on it. And it's just a really, really remarkable difference in the size of these two chromosomes. Why is the Y chromosome so much smaller than the X, right? Why is there such a big size difference there? Well, part of it has to do with the fact that there's not two Y chromosomes during our normal biology, right? So what happens is that during DNA replication, a lot of times mutations will happen, and those mutations can be really damaging. In an XX individual, a genetic female, what we can see happening is we can see that if we have a deleterious mutation somewhere in an X chromosome, that can often be backfilled using the other X chromosome. So as a result, we see that usually, for lack of a better term, full X chromosomes are typically passed on to offspring. But when we only have one Y chromosome, as we do in XY individuals, during the DNA replication process, if a mutation pops up, what will happen is it usually just gets removed and there's nothing to replace it with. So we see a shrunken Y chromosome gets passed on to the offspring. When this happens over thousands and thousands and thousands of generations, we see that chromosome shrinks and shrinks and shrinks and shrinks and gets smaller and smaller and smaller. Okay. So that's probably, we don't want to go too far down the genetics rabbit hole today, but that's probably what's happened over time. Is there's just been nothing to fix the Y chromosome because there is no such thing as a YY individual, and as a result of that, we see that we end up with this situation where the Y chromosome, those mutations have just been deleted over time, and over thousands and thousands of generations, it's just shrunk down to almost nothing. If you're wondering if the Y chromosome will disappear completely, who knows? It might. It would take a very long time, though. I think geneticists are suggesting that it might take another 1 million or so years for that to happen. Between you and me, I don't think humans are going to be around that long. So we'll see. So we might die long before the Y chromosome does, but it would certainly make for an interesting situation if the Y chromosome did happen to disappear. That's a discussion for a different day, though. There we go. 10 million years. Sorry. Conservative estimates suggest that remaining genes on the Y chromosome will completely disappear in the next 10 million years or so. So it's going to take a little bit. Okay. So the Y chromosome has this SRY gene on it. And that SRY gene is going to really direct the development of these bipotential embryonic structures. Okay. So if we go back and we look at early embryonic anatomy, which, as I know, is something that we haven't really done a whole lot of in this class, but it's worth doing in the reproductive section. If we go back and we look at early embryonic anatomy, we see that embryonic tissues have these structures that we call bipotential structures. Okay. In other words, they have the potential to either develop as male genitalia or female genitalia. And if you go back early enough in development, there's no way that you're going to be able to tell whether or not an embryo is male or female. As you probably are aware, you have to wait several weeks into gestation before you can actually start to identify genitalia on an ultrasound and things like that, right? So we're going back prior to that before you can tell the difference. As early embryos, we have two sets of ducts in our abdominopelvic cavities. One of those sets of ducts is called the Wolffian ducts. The other one are called the Mullerian ducts. You can probably guess who both of those are named after. I would be willing to bet it's Wolf and Mueller are probably the two biologists who either discovered those or they were named after. Okay. That Y chromosome has the SRY gene. Okay? If the SRY gene is present, it's going to help us to determine which one of these sets of ducts develops and which one of them does not develop. Okay, so one of them is going to develop, one of them is going to degenerate. Part of this is due to the fact, or part of the SRY's power is due to the fact that there's a protein that's produced called the SRY protein. Okay? The SRY protein is sometimes also called testis determining factor. I kind of like that a little bit better because it tells me what it does. It helps the testis to grow, right? Testis determining factor. What SRY protein or testis determining factor does is it acts as a transcription factor that affects a whole bunch of other genes, okay? So you guys probably haven't talked a whole lot about transcription factors yet, but let's just go ahead and kind of talk about those. So the SRY gene, let me put up the lights just for a second here. Punch for other pens. Green. Pay no attention to the man behind the Okay, so transcription factors, we all know that when a gene is expressed, right, this is the basic, right? This is our basic explanation. This is something that you guys learned in Bio 1, right? You learned that when a gene is expressed, right, what you get is you get RNA, and then you get a protein, right? This would be DNA, right? Right, and the two processes are transcription and translation, right? We learned this in Bio 1. You probably also learned it in high school. This is what we call the central dogma of molecular biology, right? Sound familiar? Right? DNA to RNA to protein. Okay. What a transcription factor does is if we have the SRY protein, right, or testis determining factor, right? So SRY or testis determining factor. That is a gene, okay? And when that gene is expressed, what it does is it creates a piece of RNA, okay? It creates a piece of RNA, but instead of being transcribed directly into, translated directly into a protein, what it does is it goes to other parts of the DNA and it causes the expression of more genes, okay? So RNA here is going to go then to multiple parts of the DNA, right? So multiple parts of DNA. And it causes RNA to be produced, right? So we have RNA, we have RNA, we have RNA being produced, okay? And then that will lead to multiple proteins that are all different from each other. So we have proteins being produced, proteins being produced, proteins being produced, okay? So what it does is it essentially acts as a transcription factor rather than being translated directly into a protein itself. So this is a transcription factor that we see right here. Transcript factor. Okay. So it becomes a transcription factor. And what that does, like I said, is it sort of amplifies the effect of this gene because it affects all sorts of other genes on the DNA, and not just one, right? You don't need to know really more than that. There's a whole bunch of genes in physiology that act as transcription factors. This definitely is not the only one that does it, but I just want you to understand that what it does is it essentially is going to cause more than one gene to be expressed. So we see this whole cascade of gene expression from all sorts of different chromosomes, not just the Y chromosome. This can affect all sorts of other chromosomes as well. And so we see all sorts of genes being expressed. I listed a couple of them up there. Genes sometimes have funny names like SOX9 and WT1 and SF1. Sometimes the biologists who discover these genes have senses of humor. There's an important one in development called Sonic Hedgehog and stuff like that, right? You don't necessarily need to know those names of those genes, but knowing that this SRY protein acts as a transcription factor is definitely an important thing, because we see that there's going to be a lot happening in a very short period of time, right? So let's go ahead and look at what that SRY protein, or that testis determining factor, does when it's expressed. What effect do all of these proteins have on the embryo, on the ducts, those Wolffian and Mullerian ducts that I mentioned a second ago? Okay. Well, one of the things that it does is it results in the release of three really important hormones. We have a hormone called anti-Mullerian hormone that's going to be secreted. We have a hormone called dihydrotestosterone, and then of course we have testosterone as well. Okay. These are all secreted from the gonad itself. There are cells called Sertoli cells that secrete anti-Mullerian hormone. There are cells called Leydig cells that we looked at the other day. Those are interstitial cells that secrete dihydrotestosterone and testosterone. Okay, so let's go ahead and take a look at what happens to the internal anatomy here as this SRY protein is expressed. Okay. So here you have your bipotential embryo. You'll notice that the embryo is still in very early phases of development. The lower limbs haven't formed yet, or anything like that. We do have our two sets of ducts right here. You'll notice here in purple you have your Mullerian duct right here and right here. You have a bipotential gonad. So this gonad also is either going to turn into the testis or the ovary. It hasn't been determined yet. And then, of course, directly connected to that, you have the Wolffian duct. Okay, so we have the Mullerian ducts and we have the Wolffian ducts. All right. Those ducts, as well as the urinary system, all are exiting at this point via a single opening called the cloaca, as well as the digestive tract, for that matter. We haven't completely formed all of the different openings that need to be done yet. Okay. If we have a genetic male, if we have an XY individual, and that SRY protein is present, what we're going to see is the following. We're going to see that the bipotential gonad is going to first of all develop into a testis, and then we'll also see that the Wolffian duct is going to be allowed to develop, and we see here that Wolffian duct is developing. That anti-Mullerian hormone that is secreted as a result of the SRY protein is going to cause the degeneration of the Mullerian ducts, and they get absorbed by the body. You'll notice that those Wolffian ducts are connected to the gonad, and as a result of that, that's why we see the male internal anatomy connected to the gonad, because those Wolffian ducts are indeed connected to the gonad. Okay. Testosterone converts those Wolffian ducts into the anatomy that we looked at the other day. We have the seminal vesicles, the vas deferens, and the epididymis, and then dihydrotestosterone is going to direct the development of the prostate gland. Okay. So all of that internal anatomy, you know, essentially prior to the urethra that we looked at the other day, is going to be directed entirely by this SRY protein, and it's the result of the development of those ducts that are called the Wolffian ducts. If the SRY protein is absent, in other words, if we have an XX individual, a genetic female, we're not going to see that cascade of events that occurs. As a result of that, the Wolffian ducts will not be able to develop, the Wolffian ducts will degenerate, and the Mullerian ducts are going to develop. Okay? We see that the anti-Mullerian hormone that is not present is going to allow, or the absence of anti-Mullerian hormone, I should say it that way, is going to direct the development of that Mullerian duct into becoming the fallopian tube and the uterus, okay, as well as the upper vagina. So really it comes down to which set of ducts is allowed to develop, right? And we see here, if you notice, that Mullerian duct is not connected to that gonad, and as a result, that's why we see that the fallopian tube is not connected to the ovary, because they aren't connected during early embryonic development. They're completely separate from each other. Okay. So when we look at the internal anatomy, we see that the gonads are what we call homologous to each other, right? The ovary and the testis come from the same embryonic tissue. But when we look at the rest of the internal anatomy, when we look at the epididymis, the vas deferens, the prostate gland, when we look at the fallopian tubes, the uterus, the at least upper part of the vagina, we see that those are coming from completely different embryonic tissues. Okay. Does anybody have any questions so far? Yes.
So before 10 weeks is when just one of the ducts degenerates, and then after 10 weeks is when the actual gonads kind of form?
Yeah.
Okay.
So, yeah. So about 10 weeks is what we're looking at, right? They're about a quarter of the way through gestation. If we assume gestation is about 40 weeks, which is, I guess, a nice round number, it's about a quarter of the way through gestation. We see what's happening. Okay. It's also about the time when, on an ultrasound, we can start to see external genitalia as well. So let's take a really quick look at external genitalia developing. Again, the presence of the SRY gene and that testis determining factor is going to be key to determining how things develop. Okay. Again, early in development at about 6 weeks, we see that we can't really tell the difference between males and females. This is essentially what we see right here. We see what we call a labial scrotal swelling right here starting to occur. There's a group of nerve endings called the genital tubercle right here. And there's a urethral groove that's kind of formed right here. But beyond that, we don't really see a whole lot of difference between males and females. But again, as the SRY protein is expressed and as testosterone and dihydrotestosterone exert their effects, we see that bipotential anatomy develops into the male anatomy. We see that the genital tubercle becomes the penis. We see that the urethral fold does essentially close up, and the labial scrotal swelling will become the scrotum. Okay. In females, we see that in the absence of the Y chromosome, in the absence of testis determining factor, we see that that genital tubercle becomes the clitoris, and then the labial scrotal swelling, of course, becomes the labia majora and minora, and we see the development of the vaginal opening as well as separate urethral openings. Okay. So in terms of the external anatomy, we see that a lot of it, almost all of it, is what we call homologous embryonic structures, right? The penis and the clitoris are derived from the same embryonic tissue, which makes sense since they're both jam-packed with nerve endings, right? And then, of course, the labia and the scrotum are also the same embryonic tissue. It's just a question of what did it become, right? So it's kind of an interesting development that we see. But external anatomy largely is homologous between males and females. Internal anatomy, we're looking at different embryonic structures. We're looking at those Wolffian ducts and those Mullerian ducts. Okay. Any questions about that? Yeah.
So in the internal organs, you said the testes and ovary come from the same.
Uh-huh. Yeah, so you'll notice here, there's just this gonad. It's a bipotential gonad early in development, meaning that it can become either the ovary or the testis.
And the
So,
ovaries come from the
but the ducts are completely different tissues. Yeah, either the Wolffian or the Mullerian ducts. All right. Anything else? Kind of interesting, right? To see how development occurs and the similarities and differences that we see between the male and the female anatomy. All right, let me see here. Just want to take a quick look at my notes. I think I might want to wait just a little bit until we get into, well, let's go ahead and talk about oogenesis in females and follicle development, and then we'll kind of go from there. I don't want to let you guys out too early because we got to talk about it all anyway, but I also don't want to be in the middle of something and not have it work. But let's take a minute and talk about oogenesis in females. Does anybody have any questions about development? Okay. So, oogenesis, which is the production of eggs, or meiosis, I guess, essentially in females, has a lot of similarities between spermatogenesis, but there's also some differences. The differences lie largely in the timing of when things occur. Okay, so let's ignore, just for another brief moment, let's ignore the right-hand side of this diagram and let's focus again on spermatogenesis, and let's review some of our main points. We know that really not a whole lot happens between birth and puberty in males, but once puberty starts and spermatogenesis begins, we know that we have that spermatogonium, undergoes mitosis, and one of those daughter cells enters meiosis. The other one continues to undergo mitosis, so that once puberty begins, we really see that we have this kind of continual supply of sperm for the entirety of life. Again, on average, roughly 200 million a day. Okay. In females, we see that the story is just a little bit different, right? Again, we do have a diploid stem cell. It's called the oogonium. Okay, this is out up here. This is our diploid stem cell up here. And we see that in embryonic development, it undergoes a massive amount of mitosis. So you have many, many, many oogonia. Okay. However, what we see is that mitosis actually stops, right? Those oogonia don't reserve themselves to continue to undergo mitosis. What happens is that we see that before birth, they actually all enter meiosis. Okay? So if you've ever heard, you know, kind of that fact, I guess it's a factoid, right? That women are born with all of the eggs that they're ever going to have. It's completely true, right? Men are producing sperm on a more or less continual basis, but women are indeed born with all of the eggs that they're ever going to have, because those oogonia are going to enter meiosis prior to birth. Okay, they don't maintain this kind of stock of oogonia by undergoing mitosis throughout their reproductive life. They all enter mitosis before birth. Okay, so mitosis is halted, and all of them go into meiosis before birth. Okay. I don't remember the exact numbers. I think your book might give them to you, but I think I remember reading that each ovary has somewhere on the order of about 20,000 of these. So it's still a significant number, more than a woman theoretically is ever going to need in her reproductive life. But it certainly is a long way from 200 million produced every single year. But nevertheless, this is all going to happen prior to embryonic, or prior to birth. So we see that there's a lot more happening here prior to birth in females than there is in males, right? We actually have mitosis that's begun before birth in females. But once women are born, nothing really happens until puberty begins, right? And obviously that's a variable number, where it could be anywhere from 10 years to 14 years, depending on the woman, right, before puberty begins. But once puberty begins, we see that meiosis will continue. But it doesn't continue for all 40,000 of those eggs at once. What it does is it continues for one of them per month, right? The ovarian cycle, which is probably what we'll have to spend some time talking about in lab today, is a cycle that's about 28 days long. On one average, it's about 28 days long. And we see that in each ovarian cycle, one of these primary oocytes will continue its meiotic divisions. Okay? So they don't all continue their meiosis at once puberty starts. It's a very regimented sort of a thing where one of them will continue its meiotic divisions per ovarian cycle. Okay? So what we're going to have to do today in lab is we're going to have to look at that ovarian cycle. We're going to look at the development of the egg and the meiotic divisions. And it develops inside of something called a follicle. So we're also going to look at that follicle development. But then additionally, we have a second cycle that coincides with that of the ovary, and that's what's happening in the uterus, because the uterus has to prepare itself for pregnancy. And so there's this ovarian cycle where we're looking at these meiotic divisions and the development of the structure called the follicle. But then there's a second cycle called the uterine or the menstrual cycle that coincides with that ovarian cycle in order to accommodate pregnancy should it occur. So we're going to be taking a look at that as well. Okay, so I didn't get me much farther, but I think that's a good stopping point. So let's go ahead and stop there, ending a little early on our last day of lecture. Don't worry, we'll make up for it in lab. But we'll have a little bit more time to talk about it in the lab. But I want to return to this. I want to return to this concept of oogenesis. But just know that it starts, the timing is different. It starts before birth in females. It doesn't start until puberty in males. And it's a very regimented process. It does not happen all at once. And that's a really, really important concept. All right. We'll discuss the rest of the details in lab today, and we'll kind of have some models to look at right along with it, which is why I'm kind of holding back just a little bit, is because I want to show you some models as we chat about this stuff. So I want to go ahead and put it into context. If